My name is Victor Jaoko. I'm a research scientist working at Kenya Forest Research Institute. My research specialization is on bioprospecting, that is searching for indigenous knowledge application on forest products development. Basically, it's natural products chemistry. What products can we get from the trees? That is timber, that is a non-timber forest product. So this kind of research applies a lot of chemistry knowledge, where we extract the active ingredients in plants which are useful for application in other spheres of life, like in medicine, like in pesticide, like development of soap and any other product that we can get from the plants. I authored a paper last year. The paper was titled Bioactivity Guided Isolation of active ingredients from Melia volkensii, which is a plant in the drylands of Eastern Africa, which can be used in formulation or development of pesticides or biopesticides. Which could be lead compounds in formulation of pesticide or pest control products. So in this paper we used what we call a conventional method of bioactivity guided isolation. That's a very scientific term, but what we do basically is to plants have been used to control pests before, but in their crude form. So what we do as chemists is to look at the fine part of this plant, the fine chemical in this plant part, which is actually responsible for controlling the insect pest. This could be through killing the insect pest, preventing it from feeding, or repelling. So basically that is what the paper was focusing on. This paper was published in a journal called Crop Protection. It's a high impact factor journal in the Elsevier series. Melia volkensii is a tree that is a native to Eastern Africa. Basically it grows in the dry lands of dry arid parts of Eastern Africa. It does well where there's little or no rainfall. So this plant is used a lot in the drylands to green the arid and semi-region. It's very fast growing. It has very good timber, and its hardwood is termite resistant. So the fact that it is resistant against termites is what led to this indigenous knowledge. Now the natives thought, wow, this plant, why do the ants not or the termites not eat it? So that is where the theory behind this plant being used as a pesticide to control pests came. In fact, the natives in Kitui area of Kenya use this plant to control ticks and fleas in dogs. So this is what led us to investigate further about the potential of this tree to control insect pests, mostly in the agricultural field of crop protection. So basically this plant also, once harvesting is done for timber or during pruning, a lot of waste is generated. So our research was to focus on this waste which is generated, the bark, the leaves which are removed during pruning, as well as the nuts and the pulp which are thrown away during seed extraction. So we thought of how to use these waste resources for some economic viability, which was in the control of insect pests. So we studied the waste parts actually of Melia volkensii and looked at their potential to control insect pests. The objectives of this study was actually to look at the potential of Melia volkensii scientifically, to prove if it is viable, if it's working or not. So the objective, that was the major objective, to look at the potential of Melia volkensii in control of agricultural pests. So how we did this was to take Melia volkensii plant parts, the nuts, the pulp, the leaves and the nut, dried them for like 1 month. This is to get rid of all the non-volatiles, the water, so that we get the dry matter. After this, we crushed this into powder. When we got the powder is crushed to increase surface area for extraction, so that we can get into into what we're looking at or what we're looking for. So the powder was then mixed with methanol in what we call in science solvent extraction. So when it's mixed with methanol, the active ingredients which we are looking for in the plant part move from the powder to the liquid. So the methanol extracts all the active ingredients we're looking for. Once that is done, we filter off the powder, the solid and the liquid. Then the filtrate, which is the methanol now it contains the the compounds of interest from the plant. So this methanol filtrate, we evaporate the excess methanol. What we remain with is called a crude extract. So that is what we use to formulate pesticide. That is what we use to formulate, add a little water, then spray or coat in the insect diet. Then we use for testing against the insect pest. So that is the methodology we used. The motivation for this study was the fact that Africa or Kenya is a country in the tropics. We have a very temperate climate. We have a country which is warm throughout the year. This provides a very good environment for insects to thrive. Unlike Europe, where one winter comes, one snow, and all the insects, pests are gone. In Africa or in Kenya, by extension, our climate really favors insects. Insects thriving. So, and being that Kenya is an agricultural country, actually agriculture offers a lot in terms of job creation, food security and all that. But agricultural development is impacted negatively because of the presence of crop pest. Right now, if you plant maize somewhere, let's say in the western region, in 1 acre farm or even less, I'm very sure by the time you're harvesting, fall armyworm will have come from nowhere and infest your maize. The same as vegetables. You plant vegetables in your backyard. At one point before you harvest, aphids will come from nowhere, spider mites will come from nowhere, whiteflies will come from nowhere and attack. So Kenya offers a very good climate for insects to thrive, and this negatively impacts on agricultural produce that we get at the end of the day. So chemical pesticides have been used to control pests for a long time, but this has come with a lot of negative effects. In as much as chemical pesticides are very quick in action, you spray and you see the insect dead immediately. So farmers get excited, oh, this chemical is really working. But it comes with a price to pay, because these chemical pesticides have been known to persist for long in the environment, leading to environmental challenges. Also, there have been a lot of health hazards related to chemical pesticides that are used. Like they've been reported to cause cancer, to cause a lot of negative health benefits. So this has led to researchers worldwide looking at the potential alternatives to synthetic pesticides and plants and chemicals of plant origin. We call them botanical pesticides, have been touted as alternative sources of pest control products. So this is what motivated this study, to seek alternatives to synthetic chemical pesticides used in crop protection. That can we find a safer alternative way of crop protection, and plants offer a good source of alternative to chemical pesticides. In this study, the crude extract from the Melia leaves, bark, nut and pulp, which are actually a waste product, were found in the laboratory to be very active against insects. What happens with Melia volkensii is, unlike other chemical pesticides, which when you spray, they have a knockdown effect, like there's pyrethrins, which affects the nervous system of insects and leads to paralysis and eventual death. Melia volkensii acts differently. Its mode of action is it targets the digestive systems of the insect and kind of poisons the insects. So this leads to stomach poison. It also has got an antifeedant effect. Probably when the insects taste a plant which has been sprayed by Melia volkensii formulated product, it will not want to eat that plant again. And this is the essence of pesticide application. It is crop protection. Actually, it is not to kill insects. We should not apply pesticides to kill insects. These are part of biodiversity. The aim of agronomists and farmers is to protect the crop. So so when Melia volkensii is sprayed against is sprayed on crops, what it does is to prevent the insect from feeding on the crop. And once it tastes that, it affects the stomach lining of the insect, and this leads to poison and eventual death. It also leads to low fertility of the pest. So the insect pests do not reproduce further. So meaning there's no next generation. Meaning now there's crop protection against pests. For instance, I did this study and planted maize crops at KEFRI in our greenhouse. After planting, I artificially introduced fall armyworm into the maize. Of course, they will start eating. Then after they acclimatize, like a day after, because I introduced very young fall armyworm, because the lifespan of armyworm is just 14 days. Before it becomes pupa, then it eventually becomes a moth and flies away. So the most damaging stage of fall armyworm is between around 3 to 10 days of age. So I introduced young fall armyworms. Then I formulated Melia biopesticide product in the laboratory. Then I sprayed on crops, on the maize that I planted. So what I observed is that the fall armyworm stopped feeding, and their growth was retarded, meaning they did not develop further. Meaning this class of fall armyworm which were affected did not further go to did not develop further into pupa and eventual moth. And the maize that was sprayed with the Melia volkensii was healthy as maize which was sprayed with synthetic pesticides. So that is what I found out in a nutshell. I also did the same with the sweet potato. Sweet potato is a crop that is really eaten a lot in this country. It's a very good source of good nutrients, but it's affected by this insect called sweet potato weevils. So this sweet potato weevils goes down in the ground, it burrows into the ground and pokes holes and digs feeding channels inside the potato, rendering the potato non-palatable, or it's not, the marketability of the sweet potato infested with this really goes down. So Melia products, when they are drenched, when I formulated my Melia volkensii and I drenched with a watering can into the soil, it prevented the attack of these sweet potato weevils on the sweet potato. Yes, so I can say it's a good product against sweet potato also, which apparently there is no registered chemical pesticide to control sweet potato weevils in Kenya. This I saw from the PCPB website, which registers chemical products against various insect pests. Further, I purified the crude extract from Melia in order to identify which chemical in Melia volkensii is really responsible for activity against insect pests. So I did a lot of purification and trials until I got fine ingredients or fine chemicals. I could call them lead compounds, which were pure compounds, and they're equally active against the insect pest. So these compounds, I identified them in the laboratory using the HPLC, which is a machine which is used in the lab to identify compounds. Also we used the NMR, that is nuclear magnetic resonance. It's another equipment in the lab which is used to identify unknown compounds from plant or from any other source. So I used this equipment to identify the pure compounds, and these were elucidated or characterized as tusendanine, and another one was called salininolide. So these two compounds could be used as active ingredients in formulation of safer pest control products in future.